Thank you, Ivan. Yeah, fantastic. Glad to be here. Glad to talk a little bit about the research project that I just started recently. And what I'm interested in currently is, in a way, that we have like all these different animals in the animal kingdom, and they have different lifespans. And I always think if you ask a lot of aging biologists like yourself why we age, everybody gives you a different answer. Everybody disagrees. Everybody mentions the cause of aging for what they think they currently need funding for. But I think this massive difference in lifespan is actually like one of the things that all aging biologists agree on, that this is like a macroscopic thing that is true objectively, and we have like strong data on that, so nobody would debate that there are animals that live like very short and very long. But the question is, what did evolution actually do to extend those lifespans, especially like on the molecular level? And I always think about it that like evolution, like with Darwin, like is like almost at the foundation of modern biology, and but the molecular understanding of it really is like yeah, not that well looked at, right? And during my PhD, I've worked on lipids, and I'm trying now to combine those two things, or like in a way some coincidences allowed me now to combine like lipid research and looking at lifespans of animals. And for those of you who don't know really what lipids are, because lipids are like this complex class of molecules, right? Most people think about fats, but lipids are much more. They're like 400 different categories of lipids, and most of them we don't study, most of them we don't really have a good understanding of what they're doing. And in a lot of aging papers, if there's like an arrow going away and it says lipid metabolism, that's usually just like your triglycerides and your cholesterols, but it's like very, very simplified. But lipids, I mean, without lipids there wouldn't be life, right? I mean, they're important for structure and stability. They do like a lot of signaling in health, disease, and aging, energy storage. All this wiggly stuff around you is usually like fat, and then of course they're important for like transport and delivery. And we know they change with age and disease. And throughout my PhD and with Brian's lab, we worked like on using like lipids to predict biological age, with some success in like worms and humans. And Joachim had this poster on like using it like for psychological diseases. But what I'm asking now is like, how are lipids involved in maximum lifespan regulation? And I think when I started working in aging on lipids through my PhD, this data set didn't even exist. So it's really cool that lipids getting more interest of people. And that's actually a data set that I get from Vera Gorbunova's group, and Craig has been like collecting that. It's like this data set of like 34 different mammals, like with mostly rodents, some bats, some hoofed animals, and in total, like they have like 390 lipids with like a very high quality. So it's like this idea of like comparative lipidomics now. What is interesting, if you just cluster them now with a PCA, or like just do a PCA, usually we always assume that like in aging, your first few PCs should be like in a way like be like track with age. One issue we have here is like that they're all wild animals, so we don't know their age, right? So we don't necessarily know their life stage, and there's like no like nice clustering. Like you see in a way up here, it's mostly like rodents. Then here you have like some bats, but like they're like the wild boar is like somewhere hidden in there, so it's not really to dissect. But what we can do is like when you have like those PCs, that we can actually look, okay, which of them actually correlate with like maximum lifespan, and we find 4 of them. The correlations are like moderate, and interesting, one of them is actually correlating very heavily with maximum lifespan, but also like with adult weight. And if we take now like those 4 principal components and say, train the model and say, hey, based off those principal components, so based in a way out on your lipid profile, can we actually like estimate the maximum lifespan of the animals? And we did that, and it's actually like fairly, well, surprisingly good correlation with like between the actual lifespan and the predicted lifespan. If we only use the lipid profile, if we also add the adult weight in it, it's getting even better. But what is interesting that like the first 4 principal components that almost have all the weights across like different models are pretty much used in both models very heavily, and that's all like those principal components that are actually correlating with maximum lifespan. So that kind of makes sense. Then the question is, well, okay, what kind of lipids are actually like in those four principal components, right? Can we learn a little bit maybe about pathways or which lipids are like important? And what we see is like the groups that are most enriched are like phosphatidylcholines, phosphatidyl ethanol amines, triglycerides, sphingolipids, and phosphatidylserines. What is interesting, the data I'm currently working on is all of liver, and that might be because we get like a lot of triglycerides in there. Very interesting, if you ask some of like the lipid scientists, they would tell you sometimes, well, if you do things like building clocks or doing other stuff, take the triglycerides out. Because, well, they're just storage and they're not that interesting. I usually leave them in because I think there's like probably something interesting in them. Then sphingolipids, usually we always find them massively like, get strong hits on them because they're very involved in inflammation signaling, cell signaling and whatnot. And those PCs and PS as well. I think what is interesting that like, I just marked like those 4 lipids here. This ceramide 41 is like, a lot of lipid people would coin them like the dead lipids, because I mean it's like everywhere in all diseases and whatnot. Has very high hazard ratios, and interesting in our lipid clocks that were just trained to on mortality. We see these lipids as well. Sphingomyelin 41-2 is almost the opposite. It's actually like usually reducing mortality. And those two as well. So it was good to see that like lipids that like seem to predict biological age also seem to be involved in predicting of maximum lifespan across like different animal species. So then I was wondering, okay, now we have like those lipids in those principal components that seem to correlate with lifespan, that predict maximum lifespan. Could we maybe try to make sense out of it like in a network approach, or learn something maybe about like network stability of those different species, of those animal species based on their lipid profile? That's work in progress, so that's like just like a very small conceptual idea. But the idea is, I mean, we heard earlier about like that, about network stability and whatnot. And if you think, okay, what should be a network be from a long-lived or short-lived species? Probably from a short-lived species, you would have like fewer connections of edges and more on the long-lived ones to give like more robustness. Similar, like the number of connections or edges per lipid should also be higher in the long-lived species. That also like the density should be like more dense network. And in a way, the coefficients, or like the number of modules that your lipid networks has, should also like be higher in long-lived species. So I was like just looking at this and just in a way measuring like those network dynamics across like my different species. So for each species, I created like a different network. Looked at like those 4 metrics and a few other ones, like how many edges they have, the average degree, the clustering coefficient, and the density and whatnot. And what you see, all those things like go up with maximum lifespan. So it's not always like statistically significant. It's only for the clustering coefficient. I hope this is going to get better if I add like more species that I'm going to get soon, the more data of it. But at least like I think in theory the idea is the correct one, that like those parameters like should increase with like maximum lifespan. But if it fully holds, then also like across like which clades of evolution is to be seen, because I think on the lipid profiles it's always, well, you have like, especially in wild animals, right, the nutrition matters. You have the part that is actually really the animal. There's age, so lipids are usually like very noisy and convoluted. But after all, I think we can learn something from those lipid networks. You probably should analyze them, and it's probably going to be useful. So the key takeaways are that like you have like maximum lifespan signatures that are contained in the lipidome. Lipid categories relevant for aging are relevant for prediction here as well. And long-lived species seem to have more robust lipid networks, which probably allows them like to live longer. And with that, I'm wrapping it up. And like the people who helped me with that is like Brian, Jan, Craig, Vera, and Andre. And if you like this conference, just like another plug, we're going to have a physics and aging biology workshop again at ARDD. Morten, who's I think the tallest person here in the meeting, I'm not even sure, he might be currently the tallest person in Singapore, who created ARDD and lets me run a physics and aging workshop there. We're going to discuss more that, build this community further. So I hope to see some of you there. And with that, I'm wrapping it up. Thank you. I guess we have time for one question.
Hey, that was really cool. I know she also did metabolomics, so it'd be cool to see what you get if you get the data from her with that. But
I think they might have more data as well.
Yeah, they did. But my question was, so I assume it's just changes in levels that you were looking at. But were there any lipid species that were absent in either the really short-lived or the really long-lived animals? I don't know if you could see that.
Not fully specifically, but I haven't also like really analyzed them full properly yet. I am on that.
Cool. Thank you.
I was wondering what type of animals are in there. Do you have animals that like live in water, or like the environment? How does that interact with the?
So it's mostly rodents, a few bats that fly around, and then I think it's a cow, a boar, a rabbit that are like the hoofed animals.
I see.
I think I've got data for bowhead whale as well, but it wasn't like in the analysis yet. But yeah, obviously it would be cool to see what the dolphins and like all this other.
Because there might be very different types of metabolism, right? If you're in a low oxygen environment, maybe the naked mole rat would have very different lipids.
Yes.
And also maybe what they eat, right? If you eat other animals, or if you eat grass, I don't know, there could be differences.
I believe so, and that's probably the problem with comparative biology on a metabolic level, right? It's like, well, comparative genomics, well, there's not too much changing in a way, right? You get the genome and they're all the same. But I think on the metabolomic, I guess, like, if you have like a bat that's flying around here in Singapore where it's warm and nice all the year round, and a bat in France, it's like, it's different.
And just to follow up, do you have like unique metabolites? Because there could be species that have unique metabolites.
I haven't fully looked at that yet. I would assume so. There are like some. I mean, to be fair, those are the 390 metabolites. I got like a full list of 40,000, and some of them don't even have names. So we have no idea what they are. They're literally called recon something something. They seem to regularly show up, and but we don't know what they are. Perfect. Thank you.